I want to welcome everyone. Good morning. And we are about to start our online workshop on innovation and knowledge transfer. My name is Filipa Souza. I'm a project manager at F6S, and I will be your moderator for this session. So first of all, some housekeeping rules. So this webinar is being recorded, and because of GDPR rights, all participants are muted and have their video off. So the way that all the attendees have to interact with us is through the Q&A box at the bottom of your screen. So we will not have intermediate Q&A sessions, only one at the end, where we will read your questions and your doubts, or even just your comments to our experts. Okay, so after this brief overview of the agenda, so we will have Ivana sharing with us all about SynHealth. Very briefly, it's just an overview. She is a project manager and part of the coordination team of SynHealth, the project that is represented here today. So we will continue with Elodie Naveau, followed by David Silvestre, and then Pierre Close. And as I referenced before, the Q&A session will be held at the end of our workshop. Okay. So I already kind of presented the expert speakers, and they will have the opportunity to share with us the knowledge on these areas of innovation management and also knowledge transfer. Okay. So first of all, I will stop sharing, and let's start with Ivana. Tell us everything about SynHealth.
So hi, I'm Ivana from Genos, the coordinator of SynHealth project. It is a pleasure to be here today. Thank you all for taking the time to join, and especially to our guest speakers who will share their expertise with us. Sessions like this are always a great opportunity to learn from each other. I'll now give you a short overview of the SynHealth project and how we are addressing innovation and knowledge transfer in the context of personalized healthcare. So SynHealth is a European coordination and support action focused on transferring research findings, specifically in our project glycan biomarkers, from the lab to the healthcare system. It brings together research, academic, and industry partners from 5 countries to build a foundation for introducing personalized preventive healthcare in Croatian and Slovenian regions. Our project is driven by 5 strategic objectives that shape all our activities, from AI tool development to field demonstration and building innovation capacity. These objectives are put into action through 6 work packages, each with a specific focus. The 7th work package is focused on project management, ensuring seamless coordination, progress tracking, and effective collaboration across all partners. So we are working to bridge the gap between scientific research and real-world innovations. This means transferring knowledge in a targeted way, improving market readiness, and connecting with regional ecosystems to make sure our solutions are both impactful and scalable. So what will SynHealth deliver? SynHealth will deliver tangible outcomes, validated biomarkers, prepared ERDF proposals, and AI tool to scale personalized health insights. Strong bridge between research, innovation, and healthcare policies. To conclude with this final slide, I would like to give you a brief introduction to what awaits us in today's webinar and highlight the importance of this topic for our project. Innovation and knowledge transfer are not standalone activities. They are closely connected to everything that we do in our project. As I already mentioned, within the work package 2, we are working on valorization of results from previous Horizon project and preparing regulatory guidelines for market entry. At the same time, through other work packages, we are strengthening our partners' capacities via trainings on innovation, knowledge management, and technology transfer. Our partners from University of Liege and Karolinska Institute are also supporting us with their expertise and experience. Partners from University of Maribor are developing AI tool that will help us scale our personalized health insights, and glycan age play key roles in connecting research with the market. Thanks to this webinar, we have the opportunity to further expand our knowledge and strengthen the capacities. And one more time, a big thank you to our speakers who will share their practical knowledge and tools today, and helping us ensure that what we are developing doesn't remain with academic circles, but also reaches real end users. So thank you all, and speakers can start. And Filipa, please, can you continue?
Thank you very much, Ivana. So our first speaker of this session, our invited speaker for this session is Elodie Naveau. So she is an IP manager at RISE, the Knowledge Transfer Office of the University of Liege in Belgium. And she is our specialist in intellectual property, mainly patents, being qualified European patent attorney also. So alongside with the team of RISE, Elodie is instrumental also in advising researchers on crucial IP strategy, assessing the patentability of their results, and also helping out translating all of this into the real world scenario. So thank you for accepting our invitation. And now the stage is yours. Thank you.
Thank you, Filipa, for the introduction. So I have the pleasure to speak about patents today with all of you. So welcome to this seminar. So first of all, I wanted to start with a question. I have a patent. Can I use this patent? Can I use the invention if I have this patent? Well, maybe you think, yes, of course I can use my invention since it's mine. But then you may think, well, if she asked the question, maybe it's not so obvious, not so straightforward. So my patent is about coffee capsule made of biodegradable plastic. So can I sell those capsules? Well, not necessarily. For this, you need to check patent databases. Because if you check patent database, you may find that Nestlé has a patent on coffee capsule. And they describe coffee capsule made of plastic. So just made of plastic, they do not specify which type of plastic. So if I have an invention specific on biodegradable plastic, I am still falling into the scope of Nestlé patent. So what I need to do is check if the patent is still in force and valid in the country of interest. And if so, then I need the authorization from this owner. So the patent right is a right to prohibit from making, using, offering for sale a patented product or process without the patent holder's consent. So that's important to remember that it's like a negative right. You can prohibit others from doing the same thing. It's a right which is limited geographically, so it's in the country where the patent is granted, and limited in the time since it's maximum for 20 years. Exploitation rights on a patent can be given through a license agreement. And this is typically what we do at universities, since we don't exploit ourselves, but we exploit through a license to a company. So I will give an overview of the patent procedure. It will be quite fast since I don't have much time to explain it, but anyway, it's just to give you the main things, and then you can by yourself then go further into the different steps. So as a first step, the priority filing, you can choose either national route, regional route, or international route. You get a priority date, and this means you have the right during 12 months to obtain protection in other contracting states for the same invention. It's an important stage because you can then describe the invention, and afterwards, if you benefit from this priority right, you cannot change the invention. During this period of 12 months, you receive a search report, and this is an opinion of the competent patent office about the patentability of your invention. Typically, the patent office will cite relevant prior art. Based on this opinion, you can decide if it's worth to continue the procedure. At the end of the 12 months, the priority term, then you choose other contracting states, and optionally, you can go for the PCT, which will delay the choice up to 30 months. Another important step is the publication of the application. This happens after 18 months. It means that it will become public in the patent databases. Note that what you find in the patent databases are not necessarily accepted patents, since the patent at this stage is not yet granted. It's also the stage at which a provisional protection may start. If you went for the PCT application, the choice of the countries is delayed at 30 months. Then in each country, there will be an examination. And this starts upon paying the examination fee. And the average duration is between 3 to 5 years. And also you need to pay a renewal fee every year. So each country does its examination, and each country will decide to grant the patent or to reject the patent. At this stage, you need to pay the grant fee and then continue with the renewal fee. At the grant, you also have a new publication. In Europe, after grant, you need to decide which countries to validate. So up to the grant, you have 39 contracting states in this European system. But when you validate, you need to choose which country you continue. So for that, you need to do translation and pay again the fees. There is now the European patent with a unitary effect. For 18 countries in the European Union, you can have only one validation. But still, you have 21 countries where separate validation is still necessary. So it's not really a simplified system, this unitary patent, but at least these 18 countries, so it's in dark blue on this map, have a single validation and no translation is necessary anymore. Also in Europe, you have after grant 9 months of opposition period. So it means that third party can, before the European Patent Office, oppose the patent. After this period, opposition to a patent is only possible at a national court. The maximum duration of a patent is 20 years. And for certain medical products, you can get an extension of 5 years, but that's really the maximum duration. From the date of grant, you get the effective right. So remember the right to prohibit others from making the same invention, with a retroactive effect from the publication of the application. Of course, not all the patents last for 20 years, so often the patent lapses due to non-payment of the fees. I spoke of patent databases in the beginning, so also an important thing in the database is that you can check the status of the patent. So that's the whole patent procedure with the different important periods. And an example of database is EspaceNet. And so I went to Espace.net and just I typed glycan biomarker to find an example which is in the field of this European project. And then I found this example. It's from Stanford University, detecting cancer stem cells using a glycan biomarker. So I don't know if you're already aware of this patent. I'm not a specialist in that field, so I will not go into the detail of the technology, but just my point is to show you all the information you can find in the database. So, for example, you can find the name of the applicant, the name of the inventors, classification. That's a way patent is classified, and it's another useful way to find patents in the same field without the same keywords. Then you have the different dates, and this reminds you of the timeline. So here the priority was in 2017. 1 year after they filed a second application, we see that they went for a PCT because WO means world. And it was published. So 18 months after, you see July 2018. Also, we can see that this patent was granted in Europe. So you see here the EP means a European patent. When you have the letter A, it means that it's still an application. But here we have an example with the letter B, so it is a grant. And by checking the status, we can see that indeed the patent has been granted in 2023. Besides this country, it's active in the US, but no other country besides Europe and US. So you can download the document from this database, and I invite you to go and have a try then after this seminar to find other patents. So I will come back on this example after a few words about patentability. In Europe, to obtain a patent, you first need an invention. And an invention is always a technical solution to a technical problem. It must be new, and new means that you should find at least one difference with the state of the art. Then it must be inventive. Inventive means that it should not be obvious for a skilled person. A skilled person is someone that has read all the literature in the field, but this person has no imagination. So this person can apply directly what is taught in this literature, but cannot combine in an inventive way the documents. And lastly, it must involve an industrial application. So the last one is easier to achieve. So about novelty and inventive step, knowledge of the state of the art is key. So usually as a scientist, you are used to read the scientific literature. So often at university, we have inventors which are well aware of the state of the art, but you need to pay attention to your own disclosure because this is also state of the art. So very often we meet examples where the invention was already disclosed in an abstract for a conference. So an abstract is only a very short text, but if you reveal your invention in the abstract before the date of filing of a patent application, you cannot obtain a patent anymore. So, very important, the invention must remain confidential before the date of filing of the patent application. And you can make use of non-disclosure agreement, in short NDA, which is a way to keep the confidentiality when you discuss with a partner. Now, a word about patent claims. There are two main categories of patent claims. The first category is called the entity. So you can claim a product, you can claim a composition, you can claim a kit or a device. So an example in your case would be a biomarker. We then describe this product by its essential features. So typically it will be a chemical structure or it will be the components of the product. And the second category is the activity. So we can claim a method, or we can also claim a use. So both the method and the use, in fact, it's all an activity related to the product. For example, a method for detecting cancer. In the case of the method, you need to describe which steps are performed in the method and which steps are needed to obtain the result. So I will not detail here, but in Europe, some methods, when exercised on the human body, are excluded from patentability. So for example, a method of diagnostic, if you perform the diagnosis on the body, it will not be possible to patent it. But we can still patent method of diagnostics when it is performed on a sample which is outside of the body. So I come back to the example which I found on the patent database, detecting cancer stem cells using a glycan biomarker. So this was described as a method. So I copied the claim 1 of this patent, and it's the patent, the claim as it was granted by the European Patent Office. So it's a method for detecting circulated cancer stem cells in a biological sample. And then you see that we will describe the steps of the method. So here we have, first step, we have an interaction between a biological sample and an antibody. So we speak of a sample because that's also a way to emphasize on the fact that it's not on the body. And then we have the step of analyzing a cell population of the sample. And then we specify how we analyze with two other steps. So we determine a presence of the cancer stem cells by detecting binding between the antibody and a glycan biomarker. And we specify which is the structure of this biomarker. So you see here the structure of the glycan biomarker. And then we detect a level of the cancer stem cell within the cell population, which is responsive to the detection of binding between the antibody and the glycan biomarker. So how was this analyzed by the Patent Office with regard to the patentability criteria? Well, was it considered new? Yes, because there was at least one difference with the prior art. And so this is what I found in the search report by the patent examiner of the Patent Office. He said that the glycan structure defined in Claim 1 are different compared to the prior art. And is it inventive? Well, in the search report, the examiner considered that it was not inventive because the prior art already shows that circulating tumor cells in certain cancer display the glycan structure, which is the same as one of the blood group precursor antigen. So I hope that this is, it speaks to you. It does not speak exactly to me as I'm not in the field, but I hope it does to you. So this was the opinion of the examiner. But then the applicant argued against this. So the applicant came with the argument that a skilled person would not look at the glycan structure in circulated tumor cells because these are different physically from the stem cells. So they indicated that this physical difference makes it that it's not evident, it's not obvious that we obtain that the same structure would work for different cells. So they said that the use of the claimed glycan biomarker was surprising due to these differences. And the word surprising is very important in the European procedure. So if you can show that the effect you obtain is surprising, then you can convince the examiner. And that was the case in this example. After this argumentation, the examiner recognized Claim 1 as inventive. So I will finish with a few advices on IP management. It's important to pay attention to who generated the idea. often it's the code generation of an idea. We work often in consortia, so we have several inventors. But how do we decide who is the inventor? It's important to document the creation of the invention. And then who is the owner? The employer owns the IP right. So you as a researcher, you keep the right to be named as an inventor. So it's important to document your ideas by using a laboratory notebook, either paper notebooks, or you can also use a computer notebook, as long as we can find the date at which you had the idea. Also, it's important to have consortium agreement before the start of a project. And once you get results that you wish to protect, then have co-ownership agreements. Also, use material transfer agreements. If you work with an external partner, you transfer material to this partner, it's better to sign this agreement before. It will specify which ownership is linked with this exchange of material. And lastly, the license agreement will be useful when a company wants exploitation. And so my colleague then, David, will speak more about the licensing. Thank you for your attention.
Thank you so much, Elodie. Very interesting presentation. Let's see what questions you have at the end of all the presentations. Now I will present David Silvestre. We are delighted to have him today. So he is a knowledge transfer officer also at PRISE. And in his current role, David is a vital resource, assisting researchers with all aspects of translating science into real-world solutions. So from strategic project planning and establishing crucial industry partnerships to supporting IP protection. So please join me welcoming David. Go ahead. The floor is yours.
Yeah. Thanks for the introduction. Thanks for the invitation. So today, during the next 30 minutes, I will try to convince the audience why you should engage in knowledge transfer. Oh no. Wait, my computer is not already. Oh. Yeah. So this is a little bit a call of action for researchers, and especially, I mean, I'm also addressing the young researchers. So as everybody heard about what is called the future of the European competitiveness, which is also known as the Draghi report, Europe is not doing well when it comes to translating the research they generate into innovations. Around 1/3 of patents are used for some kind of application. Although, I mean, Europe in general is doing well at the global level when it comes to the number of patents. And then the third point is, for instance, just to give you an idea, is that US startups that may benefit more from the IP generated on ERC funding grants in Europe. So we need to improve and do more with the knowledge we generate. So my presentation will have two parts. One part is going to be a little bit focusing more on patenting, and what it's more not on the formal side, as Elodie explained, but more why patenting? What is the use of a patent? You have to understand that a patent is a tool. It's not an end on itself. It's something that you think of if you want to do something with it. So it's just to try to explain why you should consider patenting and why not. So why patenting? Just to short answer, it's because when you do your research, you want to use your research to make solutions to problems out there in society. That's why you patent. So as I was saying, a patent is a tool, let's say. So, and how can a patent help you to create these solutions? So basically, it could be knowledge transfer because it facilitates collaborations with industry. Industry likes to collaborate with research projects that are already patent protected because it's something they can grasp, they can have a license on it, they can own, and they are more or less convinced that no one else can have it. Direct economic impact, it's startup creation. I think Pierre is going to explain a little bit his journey in that respect. So typically, I mean, especially in the biotech field, I mean, in adjacent therapeutics, the creation of a startup is basically the granting of a license on a patent that exists on our university. And also it helps you to get funding because, I mean, university industry likes, as I was saying, to collaborate and to license technologies from universities that are already patented. So Elodie talked about what it's considered when a patent is not evaluated, it's granted. And what I want to stress here is what is not evaluated for patentability. So just to sometimes researchers, when they patent things, they have some expectations that then when they confront to reality are not met. So when you patent, even if your patent is granted, the patent doesn't say anything about the level of maturity of the invention. It doesn't say if your technology is ready to go to the market or you still need to work for the next 20 years. That's nothing about it. It also doesn't say anything, I mean, or very little, how likely is the invention to solve a problem? I mean, how is it technically super difficult to do it? There are lots of competitors. Maybe the problem you want to tackle is really complex, might change and stuff like that. That is not on the patent. And also the third point, the patent doesn't say, it says something about the novelty, the inventiveness, but it doesn't say, I mean, what's your competitive edge with respect to all the things that are out there, because it will always have competition when you have a new technology. It's just something you always have to keep in mind. I mean, how better my technology can do with it's already out there. And that's something you have to find out for yourself. I mean, the patent will not tell you that. Another common thing is, of course, researchers, they are used to publish. That's what they are being paid for. They are typically, they're trying to extrapolate patents with publications. There are many differences between patents and scientific publications. Here it's a short list, let's say. The purpose of a patent is basically, it's a negative right, as it's called. It's prevent others. You have your monopoly on your technology. You prevent others from using it. So a scientific publication, it's the opposite. It's you want just to share so everybody can use it, everybody can profit from it. And the patent, the focus on the content, when you read a patent are not easy to read because it's very focused on the practical applications and technical details. So, and sometimes it's also focused on hiding those. Whereas a scientific publication is the other way around. You want to make people use it, understand it, make it clear. The granting procedure of a patent is rather complex. You need an expert like Elodie to do it. You cannot do it yourself, or I wouldn't advise it. Whereas a scientific publication is rather straightforward, let's say. So they try to make it as easy as possible so everybody that has a good piece of science can publish it. The costs are quite different. Scientific publications, it depends which journal you go for. Typically, I mean, can be on the scale of like thousands of euros. Patents can be really, really expensive. That depends a lot of the patent. But if you just start having discussions with evaluators and you have just to then contract like lawyers and people like that just to help you have your patent granted, then prices can escalate pretty quickly. Target audience, patents is industry, people that want to exploit them, scientific publication research. Scientific publication has not legal implications. So if you publish something, I mean, you can retract a paper and you are not going to have any legal implications of that. Whereas a patent, someone can take you on court for this. So you have to be careful what you say on a patent. And just to final, also an important thing is that the maintenance of a patent, as Elodie was explaining, you have to pay the annuities every year. So you need to understand how to do it, how that's been done. So you need someone that really knows how to do it. Whereas a publication, it's published, it's out there, and that's it. You kind of like don't think about it anymore. This is an important point. So when you think about to patent or not to patent, this is typically in a TTO what we evaluate when a researcher comes with a piece of science, say, okay, we want to patent this. So this is like typically the things we're going to evaluate. So for instance, the problem, the problem, I mean, so you define a problem, so you already identify what is the thing you want to solve, and that's eventually, that is quantified. So you can, with this technology, do better than something that is there, something, I mean, you can grasp, you can really define. Typically researchers, they don't think on that terms. So it's like typically this expression of technology push versus market pull. So maybe you like a lot your technology, but what you have to think first is like, what is the problem I want to solve? So that's really, really important. When it comes to maturity also, so the invention has to have some level of maturity, has to be evaluated in a relevant environment, and it has to be just an idea. Confidentiality, as Elodie was explaining, the resource has been confidential. If you just any kind of disclosure, you go to a conference and you present a poster, that cannot be patented. So having just keeping your resource confidential is really important. Another point is that so also another thing we evaluate also is like if you have a plan on how to want to do it, because I mean, okay, you have a technology that might be interested, you patent it, but then you don't know what you're going to do next. So you should already have an idea. You don't have to have like a development plan, let's say full blown, but you need to understand how you or someone else could do it. You need to have at least a general understanding of it. And also, I mean, if the inventors, the researchers are implicated on that process. So that's a really important thing. And typically licensors, I mean, when industry it's interested in a collaboration or a license or a technology that needs further development, they want to see that. They want to see that the researchers are willing to participate on the journey. And also the market, I mean, a patent, it's meant to put something on the market. It's for an industrial application. So you need to understand what is the market. I mean, there's a lot of money to be made. Typically, there are two sides, two numbers that industrials want to know. Market size and what is called the compound annual growth rate. That means how it's going. Today, there's an interesting market, and this market is going to grow in the future. So you don't need to be an expert, but you need to at least have in mind those numbers. Another challenge in academia is that you are being paid and you are being encouraged to promote what you're doing, to disclose your research. But that is contradictory with patenting. And at the same time, an invention should be mature enough at the time of filing, as I was saying. So we cannot, that means typically that's a challenge in TTOs like ours. We cannot prevent the researchers from doing these disclosures for a long period of time before patenting. So the fact, and that's a common thing in all TTOs, in all universities, that the patents that come from universities, typically they tend to need significant maturation. So what we advise to researcher is try to be selective on your disclosures. If you think that your invention can have an industrial application, just be selective on your disclosure. Try to keep it secret as long as you can. And there are workarounds for that. Yeah. For instance, PhD defenses can be made confidential. They're still accepted by many doctoral schools. And when you want to disclose in conferences, you can just like, okay, what is important? What is information that is going to be interesting for the audience? And it's like a nice to have. What can I show and what I can keep secret? And why this is important? I mean, and at which point in time you are going just to patent? Because as Elodie was saying, once you do the priority filing, just the very first filing you do, typically you have 20. The lifetime of a patent is 20 years. And when you think in relationship with the timeline of how long it takes to develop a drug, it's about 10 to 15 years. So that means that the commercial life of a patent in the therapeutic space, in any case, is around 5 years, 5 to 10 years, best case scenario. So that means if you patent way too early, when you start developing something, I mean, the patent is maybe not going to be interesting. Typically, a patent that has like around 10 years, we can go pretty quickly. It's not that long. And and it's not like a straight path to a concrete product. We tend to think that patent will be unlikely give rise to a product. So that's very important, just to try to patent as late as possible. Or at least, I mean, when you really have like a decent data package and the technology is mature enough. But we know in academia, this is really difficult to do. Because what happens is, as I was saying, a patent has like a typical limit. The exploitation type of a patent can be between 5 and 10 years. And when that patent it's over, let's say, the product, it's off patent at its core, and anybody can do it. So it becomes a generic. And this has in the industry had this dramatic name of patent cliff. So that means that companies that put this drug in the market, they lose this income. So this is just to give you a flavor of what the different what are the different products that will be off patent in the upcoming years, and those are in $1 billion. I mean, how much money less the companies, the pharmaceutical company, would just stop having. So that is just very important to have a patent active in the market as long as possible. So that was the first part when it comes to patenting and why is it important. So my take-home message is patenting. It's as I say in mathematics, it's necessary but not sufficient. Okay, so you need a plan. You need to understand. You need to know what problem you want to solve and how you're going to solve it, and then the patent comes in. Then my second part of the presentation is like when I explain this is to a call of action. Let's say for the people here on the audience to engage in knowledge transfer in Europe. There's a huge need in academic settings to have more researchers getting involved in this journey. So why knowledge transfer? I mean, why you should think about it? Because that's first, number one, that's the most important, because you are really enthusiastic. You really want to solve a problem. It's like it's like some people say, fall in love with the problem. It's like you see something there, say, this is something, I mean, I could really help to improve and I can do something about it. And maybe getting rich and famous on the way, that's also fine. But you will not get rich and you will not be famous if you don't solve a problem. The second one is career prospects. I'm going just to discuss a little bit about it. And also third one, even if you want to stay in academia, I think engaging in knowledge transfer, it's a positive loop that will help you even go further with your academic research. So talking about the career prospects. So a PhD today is a structure. I mean, PhDs, a structure of PhD comes from Germany, mid-19th century, and it's still basically the same. So it's basically a PhD student and a mentor and a one-to-one relationship. And everybody knows that this completely changed nowadays, much more complex, let's say. And a PhD advisor, it's not sufficient, let's say, to have a successful PhD. There's a whole community around it that it's needed. And then, I mean, those are numbers in the US, which in Europe it's more or less the same trend. 60% of fresh PhDs in the US, 10 years after they have their PhD, have a permanent position. That number dropped to 54 in the 70s and it's 38 in the 80s. And here what you can see, it's like this is a cumulative number of total PhD through time, and this is like the cumulative number of total positions in academia. So basically, it's really, really hard to find a position in academia for a PhD nowadays. So what are the consequences of this? Basically, there's a kind of an exodus of people from academia to industry that has different names. And this is a tendency, and it's everywhere in Europe, in the US, in most industrialized countries, it's a fact, let's say. So the thing is like there are not sufficient stable positions in academia for all the PhDs we are generating. So how does knowledge transfer would help you in that respect? So it will put you in contact in collaborations with industry. You will learn really a lot by collaboration with industry. I mean, it will just open you the door to a new world. It will help you also training in entrepreneurship. If you want just to do it yourself, you want to create a company and you want to do something, I mean, that will give you the tools to do it, just to say, okay, I want just to, my PhD, my research project, I will tailor it in an application. The networking, it will just put you in contact with a lot of people with different backgrounds, different skills, and so on. And that's very useful. It will give you access also to training. There are lots of trainings related to knowledge transfer nowadays, which is IP, licensing, also project management, and so on. And also, I mean, if you do the jump from academia to industry, I did it myself, it's a jump into the unknown to a certain extent. And if you already know what to expect, that will reduce the risk to have by surprises when you do this transition. So that was from the perspective of from the career prospects. So then more like for the people that want to stay in academia and want just to keep doing research in academia, why engaging in knowledge transfer? So there is I'm not going to enter here into the details, but there is like the idea that there is industry, or mainly big industry, big pharmaceutical companies, they do less and less research, R&D, and that they externalize that. So they externalize that, but there is the consensus that there is a need for new technological innovations. It's a paper from Nature in which they show that patents and also papers are becoming less and less disruptive over time. It's all cumulative, but less disruptive. Everything that new indeed, it's always building on something that exists previously. So we are not finding like disruptive solutions to the problems we have. And there's a need for people coming just really thinking out of the box with new ideas and new ways of solving these problems. And the industry relies more and more on academic innovation of SMEs. It's really, a tendency just to outsourcing innovation in academia and small structures from big industry. This is just a couple of numbers in which almost 50% of technology transfers that means technology being acquired by big industry, indeed, is coming whether by academia or SMEs. In the biotechnological field, SMEs, oftentimes, they are spin-offs for universities. So I think, we can mix both there. So there is a need for these solutions, and these solutions by big industry are being looked in academia and SMEs. So how big industry does that is this concept of open innovation. There are books reading about it. I'm not going to enter into the details, but basically that means that at different stages on their development, product development paths, industry will scout and incorporate different types of solution that they will just mix or as I was saying, incorporate it on their products. And they have like all big pharmaceutical companies, they have their open innovation programs in which they have their call applications in which you can just apply for them, propose your idea. And then if you are very lucky, because the attrition rate is incredibly high, you can participate on those. So there is really an interest from industry to collaborate and to find these innovative and potentially high value ideas in academia. So how open innovation looks in real life? There are three types of of ways in which it works. It's licenses. Typically, if you have a patent or your innovation, the company will have a right to use your patent, typically an exclusive right. So for instance, you develop a biomarker, which is the case here. So company says, I want to develop just to put it on the market. They will do it themselves, or they can do it in collaboration. Two nice examples are the COVID vaccines. One was with a German SME at the time. Now it's a big one, Biotech and Pfizer. So basically Biotech what provided was the technology and Pfizer what provided was like the production and commercialization. And there was a similar agreement with the University of Oxford and AstraZeneca. And another type, which is more related with small companies, is called mergers and acquisitions. So big industry, they're like companies, small companies that already have a product which is quite mature, ideally in clinical phases, and they just buy the company, or they just merge it on the structure. And this is a list of recent mergers and acquisitions. Big companies buying or merging with smaller structures. So that's a little bit exaggerated, I would say, but I think, there's really, you have to be aware of that. I think researchers are not aware of that. The industry is really looking around. They really want to know what universities, what research labs are doing. It's not for free, of course. It's not easy, but they are looking this sweet spots, let's say, in which their needs, what they're looking for, could be potentially solved by the research that is being doing at universities. And then just to finalize, so what I want to, because researchers that are in academia say, well, I don't want to go to industry. I can do it. Not, it's true, it takes time. It really have to, you have to change your mindset when you collaborate with industry or you are engaged in knowledge transfer. You need to focus. You cannot start collaboration every 6 months. You have to be careful about your disclosures. You cannot go to a conference and just to talk with people about everything. It's true. So it needs to change a little bit of a change of mindset. But I think, I mean, there's a lot to gain by researchers that wants to stay in academia by engaging on this. First of all, it will increase the visibility and impact of your research and also the reputation. It will give you access to resources, networks, and idea. Potentially, I mean, one of the best scientists I found, I saw it in some industries, let's say. It's incredible. You would be surprised how knowledgeable, how much they know, how sharp they are. I mean, you will be in contact with people that are really, really, really good. You can also learn from them, because when you learn how a product is developed, it's all about the data. At the end of the day, it's all about the science. That's what leads, of course, there's a market considerations, but science, the data, it's on the lead. So you will learn also for a scientific perspective. It will help you to do better your I wouldn't say better, but to think in a very deeper way of the things you do on the lab. And also, last but not least, I think it's maybe a neglected point. It will help you boost your research, because by doing these collaborations, tools will be generated. And then you can use these tools. For instance, I mean, if you are collaborating on a drug development project, this drug, then you can use it as a tool. And potentially you will be the only one, or in any case, the first one to use that tool. So that will, again, help you increase the visibility of your research, have more impact, and have better publications, and so on. Because, and that's, it's complicated. It's really complicated. That's why we don't hide it. This is, again, on the drug development space. It takes a lot of time. It takes a lot of expertise. It takes a whole town, let's say, to do it. And you should be aware of it. And this complexity, I would just say 2 words before I run out of time. Just 2 concepts I would really like just to make understand. When you engage in knowledge transfer, there are 2 concepts, the concept of technology readiness level and customer readiness level, that all researchers should be aware of. TRL, that comes from NASA. They were developing the rockets. There were different projects internally, and everybody was talking from different departments, and no one knew, there wasn't common information about how far these rockets were in development. So they came with this idea to TRL. So 1 is just for the idea, and 9, it's already the thing already done. So they can communicate in the levels of TRL to know which was the level of development. And that's a little bit the standard nowadays in all projects when you want to develop a product. So when you go to engage in a drug development path or any kind of product development path, this is something you should always be able to explain how mature is your project in terms of TRL. That's a technical part. Same concept is for the customer readiness level, but that's related of how likely, how much you know that your solution will solve the problem. I mean, how close you are to the market. And that's typically the researchers that tend to neglect this. And both go in parallel, and both are as important. So when you talk with industry, they always want you to know, to hear from you, that you know in which state of a TRL your project is, and ideally also your CRL. CRL is not obligatory, because maybe you're still thinking commercially, but it's useful too. When it comes to TRL, and this is the reality also from academia, there is one, and this is like we are confronting of TTOs every day to the value of death. So basically, academic projects, they are early TRLs, so there's public funding on it. But when you move ahead on the line, so you move more to a prototype and more close to, you move more from development instead of research, there's no funding. And it's very hard to have traction with industry, because typically the industry will say the project is not mature enough. So this is where TTOs intervene. And this is like a reality. There's a lot of European initiatives trying to solve this, basically providing grants and trying just to make collaboration with industry easier. But if you engage in a technology transfer path, you will face the valley of death. The way to be ready for it is to already think or know that it exists and try to think, I mean, what are the help you can get in terms of like knowledge and resources. And just to finalize, I want just to wrap up saying what are typically the challenge when you move from a purely academic research to knowledge transfer. First, most importantly, understand the problem. You really need to understand the problem. What is exactly the problem you want to solve with numbers, let's say? How much you want to extend the life of a patient? How much faster you think you can do it? How much cheaper? I mean, how much more people can use it? So it should be a number, even if it's a ballpark figure. And if I were to do it, it's just to talk to users and experts. Just go out, talk to people that does it. Get out of the lab. That's super important. I mean, don't leave the bench. That's another important thing in knowledge transfer. Go out on the field, confront your idea as early as possible. Same thing for evaluate your proposed solution. Just to understand your proposed solution. Okay, how likely is my solution to propose something, or is this just like a crazy idea? Same thing. Go out, talk to people, talk to experts. Don't be scared to talk to people. Early stage ideas. No, I mean, I never saw someone steal it. There are lots of good ideas out there. So don't be scared that someone is going to steal your idea. I mean, of course, I mean, the data should be confidential. You have to identify the advantage of the competitors. So you really need to prove. This is something typically the industry will ask you, okay, I mean, how much better? Just tell me who are your competitors, first of all, that you know them. And what is your USP, as it's called, your unique selling point? You should be able to say that. Have a realistic plan for the whole process. You don't need to be an expert on everything. As I said in one of my previous slides, it's an incredibly complex process. But you need to have an idea. Typically, I mean, if you are working in life sciences field, at least what is really important is to have an idea of the clinical part of it. So how patients would use it. I mean, if it's a therapeutics, how clinical 1, phase 2, 3 would look like. How would you compare it to? What's going to be the standard of care and things like that. Very top level, but still you need to think about it. And then the last one, as I was saying, the value of death. Maturation over these TRLs 1 to 4, and that what can help. It's difficult anyway. It's to have a development plan, think about it from the beginning, and just try to think. I mean, where can you get the money to move forward, and who can help you to do it? I ran out of time, so I'm not going to go through the last slide. And okay, thanks a lot for your attention, and I hope that I convince you to talk to your knowledge transfer officers if you have them, and yeah, be part of the journey in taking Europe to the next level in innovation.
Thank you so much, David. It was very important, mainly for young researchers, and I'm talking from experience. Long ago, I was a young researcher, and I was not properly informed about what happens next. So these sessions are very important to disseminate this information. So thank you. So now we will follow, we will have the pleasure of introducing Pierre Close. He is a professor at the University of Liege and also a co-founder and CSO of a Biotech that he will tell us all about. So beyond his academic achievements, he's also an entrepreneur, and he is part of a Biotech spin-off dedicated to translating RNA epitranscriptomic research into novel therapeutic strategies for treatment-resistant cancers. So we are very excited to hear from you, Pierre. So the floor is yours. Thank you.
Hello, good morning, everyone. Thank you for the invitation. So I'm very pleased today to explain a little bit the story, our scientific story that started in a lab and happened to be at the origin of a fantastic biotech adventure. So what we mainly discovered over the years is the importance of a very specific biochemical reaction touching tRNA modifications, and we open this space as a new therapeutic target space in oncology. So today I will discuss a little bit where we come from a scientific point of view and how we actually get into the business side. And to summarize a little bit this journey started mainly through a lot of work for more than 10 years in an academic lab at GIGA, University of Liege, involving a team, many collaborators, and mainly what was important at that time to promote innovation is to think about how building new models, building disruptive hypotheses, something that would be a bit out of the blue, and also what academia offers at the time is, and still now is the freedom and the ambition to do great science. And of course, the journey is, and today I'm planning to discuss how we went from the fundamental science and main discoveries to a biotech company that is now 3 years of age, which we call the THERATRAME, in which we develop first-in-class therapeutics, where we actually propose a new mode of action in oncology, and for which we actually we plan to be first in human in 2027. Of course, in this biotech company, it involves, the implementation, the scalability of new technologies, to build teams of people in the company, but also of experts around the company, and to really think about how you position your company project and your product in the landscape of all the biotechs and the pharma, and of course the strategy to fundraise. In between those two different aspects of the scientific journey, you have a lot of different works that I summarized here, and that are very important. It takes time. It also takes a lot of networking, and I would say, as a background information, is that this journey is primarily a fantastic human adventure, because in both sides, what makes the great science, or what makes this great project going on, are meeting people, and meeting people that actually want to make the difference together with you and your team. So overall, I think the time is also the time to meet the good people. So I was thinking for this lecture is to really go for a few slides on what we discovered and what was the fundamental science behind our project first, and then see how we actually transition towards this project of the biotech. So I will go back really much to the basics and of the gene expression system. So you know that genes are enclosed in the DNA, transcribed into RNA, and the RNA is translated into proteins. And for a long time, mRNA translation, so the translation of mRNA into proteins, was believed to be normal, systematic in the cells. But over the last 10, 15 years, we realized that mRNA translation step in the gene expression is really a step of high regulation, specifically in context of stress response or cancer pathways. It's very funny if you look at what a cell's express in terms of mRNA or what a cell's express in terms of proteins is actually quite different. And we like to see that, to say that the mRNA expressed are defining the identity of the cells, while the proteome that are expressed define their function. In between those two different mRNA and proteins, you have this complex and highly regulated mRNA translation machinery enclosing the ribosomes. And today we'll be talking about a particular molecule that is quite interesting in the middle. It's called, those are the tRNA, transfer RNA. And those are adapter molecules that can actually really able to read the genetic code in the mRNA and translate it into amino acid and proteins. tRNAs, they look quite essential. They look quite very important for the cell homeostasis. And in fact, when you look at the RNA, the tRNA, you look at a funny structure here, and you also discovered that there are many, many different chemical modifications that are added to the tRNA to ensure both the stability and the structure of the tRNA. But if you go down in the anticodon loop here, you see that these modifications are very important to define the way the tRNA is going to read the genetic code and translate. And this world is called the tRNA epitranscriptomic. It's called tRNA modification. And over the last 10 years, what we discovered in the lab is that these tRNA modifications is really a way to promote tRNA function and to really promote specific function of tRNAs during translation. It really promotes translation reprogramming and phenotype switching. So it means that if you look at a tumor, you have a lot of different cells doing different things, and those are phenotypes. And eventually during cancer development, the phenotypes, the function of the cells are going to change, and this is what we call phenotype switching. And you can look into this from the angle of the cancer cells, because the cancer cells are slowly able to do different things, for example, to produce metastasis or get resistant to cancer. And also from the side of the human cells, you see that there's a lot of remodeling of the immune cells in the tumor. More precisely, what we looked into here is really the tRNA at the anticodon loop here down there. And what we see is that they promote translation reprogramming. And this is something that is quite particular as a mechanism in the cells, and actually defines very much a specificity of these reactions in the cells. What we see is that if we prevent this tRNA modification, the ribosomes are having a problem during translation. But the consequences are really not what we expect. We see that most of the genes do not have a massive problem during translation, but some proteins will be specifically degraded. And in fact, when we talk about tRNA modification, we actually think about protein degradation and proteostasis. And what is interesting for us, and in the context of Biotech, is that inhibiting specific tRNA modifications here at the down part of the tRNA specifically induce cell death in cancer cells, but not in healthy cells. And we also found that this mechanism could be the source of immune regulation in cancer. Those are the primary publications, scientific publications, highlighting why we believe that tRNA modifications are so specific to disease and not very important for healthy tissues. Those are two studies using the first conditional knockout mice for a tRNA modification enzyme. So this was done in the intestine and in breast. And the funny part here was that the expression of the main enzymes responsible for this anticodon tRNA modification was not only poorly expressed in normal tissue and overexpressed in cancer, but in the knockout, you see that the complete inhibition of these enzymes produces no phenotype. So the mice is perfectly healthy. While when we trigger tumor initiation, we see that the inhibition of these enzymes completely inhibit tumor formation as compared to a cancer mouse. And essentially the same information was true in breast cancer. We see very low expression in normal breast, high in cancers, and very specific phenotypes towards the disease. So for us, this was the first evidence in vivo that a tRNA modification could be extremely specific to a disease and not very harmful to or very needed in healthy tissues. Voilà. In 20, so what happens here? Okay. One of the key papers that was a determinant for the Biotech project is the following paper, because we first looked at conditional knockout in the past. Now we tested and we looked at patients, and we published this paper in 2018, which was the first paper linking tRNA modification with patients, and this was in melanoma patients. And essentially what we saw in that paper is that inhibiting ALK3 actually specifically kills melanoma, BRAF melanoma cancer cells, both patient-derived, but also in mice using human BRAF mutant melanoma cells. We also found, very importantly, that this tRNA modification was really a driver in acquiring resistance to MAP kinase inhibitor and BRAF inhibitors, which is the main targeted therapy in melanoma. So for the first time, we put this tRNA modification under the radar of the patients and also probable likely drug development programs. And this is a work that is in revision, but this is also very important in this discovery pathway. In this work that was performed by a PhD student in my lab, what we found is that this tRNA modification enzyme L3 is really able to regulate the tumor immunology in melanoma. Essentially, when we inhibit this enzyme, we actually induce a very specific Th1 anti-tumor response within the tumor, which is a unique mechanism in oncology. And we also found that inhibiting these enzymes very strongly synergize with checkpoint inhibitors and immunotherapy, mainly here PD-1 and CTLA-4, which are very well known. And so in this new scientific paper here, we actually link this tRNA modification with tumor antigen presentation and immune regulation in melanoma. So those are quite a lot of information, but they all have a quite high potential in the clinics. And it was a little bit odd, back in the days, to believe that tRNA modifications could be quite specific to disease and therefore be therapeutic targets. But in fact, what we found and published over the years is a lot of evidence showing that tRNA modifications, mobile tRNA modifications, really promote cancer phenotype, that cancer cells die if you inhibit this tRNA modification. And by optimizing translation, they actually promote immune evasion and regulate tumor immunology. And so from those papers and those innovations, we actually thought that this could actually represent a good therapeutic target in oncology. And this was the beginning of the thinking to create and found THERATRAME, which is the Biotech. And so now I'm going to switch towards the THERATRAME project and how from this basic science project and papers, we actually now develop, drug development pipelines in this biotech, and how also we present this to investors or to external parties like pharma companies or other biotechs and so on. But before that, I wanted to spend 2 minutes explaining a little bit the path towards the funding of THERATRAME. I mean, this is extremely simplistic, of course, but it starts with a great science supported by key agencies. And in 2017, we actually, when we started to get some very strong evidence of this patient-derived biology, we started to think about developing an HTS, high throughput screening assay development. And so we got some bridge found, and we could, together with AXAM, try a lot of different assays to see whether we can have one. So we, over the years there, so it took 3 years, we actually failed quite a lot of time because tRNAs are not, usually targeted as drug target. And so we had to establish a good HTS capacity. Eventually in 2020, we reached a robust HTS, which was for us the beginning of the story. We patented some of the technologies, and we started to incubate the idea. Incubation is more the idea, and also a personal incubation to see how, from a scientist, you actually become someone developing somehow a business. And so for that, we got help by people, coaches, but also by the strong ecosystem here in Liege in Belgium. So after the HTS, we got actually identification of hits, which would be potential inhibitors of those tRNA modification in 2021. And this was the beginning of the story where we actually started to negotiate with the university a licensing agreement towards the foundation of our company, and this was concomitant with a fundraising. And we actually created THERATRAME in 2022 with 4.5 million, which was divided by different parties. I have to highlight here the importance of the private investors, because they really are the people giving the credibility to your project and attracting then public investors, and eventually in Walloon region, non-dilutive money, which is quite important to de-risk the first step of the project. Over the last 2 years in the company, we mainly established the technologies with a different scale, but we also developed the drug discovery pipelines extensively, and we reached an important milestone in 24 where we actually got a structure-activity relationship in a chemical series that was not only specific but also working in vivo. So for us, this has been a very important milestone, and now over the next year and a half, we are designing a strategy to go to first patients, hoping to inject this first patients in 2027. So here is a little bit the pitch that we do in the company. So from this basic science, how we actually communicate this to external parties. So at THERAtRAME, what we do is we would like to eradicate cancer by designing tRNA modulators. And the way we present this is that cancer remains a disease that is extremely difficult to treat, and also that is also increasing in populations. And today there are two main ways to kill cancer cells. One way is the development of chemo and targeted therapy, which would actually exploit the vulnerability of the cancer cells. And you also have the immunotherapy, which on the other hand exploit the immune cells of the tumors and educate them or release the inhibition to kill the cancer cells. But both approaches have big limitations. Not mainly that some of the patients simply do not respond to those therapies, and many of those develop resistance over time, which is, of course, one of the main factors promoting, defeating, let's say, the battle against cancer. And so overall, there is a need for a new disruptive standalone drug therapy that would combine the precision and the potency of a targeted therapy with the durability of an immunotherapy. And here at THERAtRAME, what we aim to do is to unlock lasting cancer cures with the development of tRNA modulators. We call tRNA modulators. Those are first-in-class small molecule inhibitors targeting tRNA modifications, and we believe that those are a new class of cell-killing small molecules that targets the tumor dependency on a specific translation. Our first target is ELB3. So ELB3 is a tRNA modification enzymes. Why is ELB3 so interesting? So I showed you some of the basic science that we published, but generally speaking, if we simplify this, we could say ELB3 activity is innervated in cancer, and it's, we believe, a very much tumor-specific target. So it is upregulated by oncogenic pathways, which are activated, of course, in cancer. And by using mouse models, we demonstrated that ELB3 is essential during development, mainly on neuronal developments, but is dispensable in most elderly tissue, enabling safe and tumor-specific targeting. And from a mode of action point of view, we found that ELB3 inhibition specifically kills cancer cells and promote a specific Th1 response within the tumor by promoting tumor antigen presentation. And so what we offer at THERAtRAME is a strategy, is a unique synergistic mode of action. We believe that the tRNA modulators targeting ELB3 would combine two important mode of actions. First, kill the cancer cells directly, and second, activate a specific Th1 response, immune response in the tumor that are, and those two mode of action synergize to promote the antitumoral effect first, but also duration of the effect. And we believe that this is a unique mode of action that is not really well seen in oncology. So far, over the last 2 years, we have developed, a lot of chemistry here, a lot of assays. We have developed compounds targeting ELB3. And we came up with a chemical series that is quite now very exciting. So is a first-in-class inhibitor for tRNA modification, ELB3. It's very selective in nanomolar affinity. It's a potent, low macromolar in cells. It's very selective to tRNA modification. And we actually made sure that these compounds display an orally druggable profile with high solubility and all the properties that are needed to develop oral drugs. So our first series here, well positioned for an oral drug development. Those are, key data showing, how well is our L3 inhibited in our cells. And not only the enzymes is actually strongly inhibited, but you see, if you look at the tRNA modification, you see a very huge drop in the specific tRNA modification dependent on L3 in cells. One of the main important results that we had in late 24 is we actually got in vivo. So this THERAtRAME is a molecule that's not only selective and potent, but it's also working in vivo. Here we looked at the melanoma tumors that are not responsive to any checkpoint inhibitors or any T-cell dependent strategies, and we see that the inhibition, but the TTR1 compounds inhibition of L3 produces a very strong and stable antitumor effect. And from a safety point of view, there was no clear side effect in terms of immune cell activation or toxicity in the main organs that we tested so far. More precisely on the mode of action, so we could see that this inhibition of L3 with TTR1 activates the T cells, the CD4 T cells, the Th1 response, as was shown for the genetic model, but also led to a very strong CD8 T cell activation. So validating our data that we see, that we publish and we will publish from the point of view of the lab. At THERAtRAME is not only developing drugs against L3, we actually built a whole platform that we call EpiFind that is based on patented tRNA-centric assays and methods and know-how. So we have 3 different pillars where we actually discover. We have screening, a unique screening approach to discover new small molecule inhibitors against tRNA modification enzymes. We have a collection of different models and know-how to validate in mice or in patient-derived system. And we have this unique software AI approach to position our drugs into indications and patients. If we look a little bit outside of THERAtRAME, so THERAtRAME is definitely a pioneer in the tRNA modification space, even though now we just heard recently that we have a couple of competitors starting targeting tRNA modifications, but also looking at companies developing inhibitors against other RNA modification enzymes, mRNA mainly. And of course, we look at competitors at the level of the clinical space. In this case, we look at melanoma, but also other indications, and we actually position THERAtRAME as compared to the main actor in this clinical space. So we have a big advantage here, is the multi-cancer potential for our drugs, and the potential to have new partnerships and clinical implications. From, let's say, a more financial perspective and value creation plan, so we got the company a 4.5 million as a seed that we just extended with another 3 million here. And with this seed extension, we actually plan to arrive into IND-enabling studies, ready to start the clinical trials, phase 1/2. That we planned in late 26, early 27. So we'll be looking for an additional fundraising over the course of 26 with quite a large amount of money to actually enter into the clinical phases, to expand our IND-enabled activities to a second indication, and to develop our second programs against enzyme B, for which we already have leads that are validated, but they have to undergo lead op and in vivo validation. So, of course, when we do such a project, you're not alone. So there are 2 important parts here that I would like to highlight. Is first, the team is a very dedicated team of young guys who wanted to make the difference and to really be part of the innovation, not only as a worker, but also as a mindset, as an attitude. So we have a fantastic young team, fully dedicated to our science and programs. And we also have the chance to be surrounded by leading experts in the field, in the regulatory affairs and agencies point of view, Guido Razi. We have an expert in medicinal chemistry, Christelle Menet. In IP strategy, Emile Pot. We have a key opinion leader in clinical activities in melanoma, Mike Davis from the MD Anderson. We have an expert in PK/PD and bioavailability of our compounds, is Manfred. And last but not least, Jean Compalbert, who is a multi-entrepreneur in drug discovery, is the chairman of our board of directors. So I would like to conclude here by saying that at THERATRAME, we propose a disruptive first-in-class approach with novel anti-cancer therapy, with a unique mode of action. And we have strong preclinical data in vivo and also safety. We have also a patented proprietary scalable platform that will enable us to target multiple enzymes in multiple indications. And last but not least, we are de-risking strongly the clinical strategy to be able to have a first-in-human in 2027. So overall, what we believe is that the tRNA modulators represent a disruptive shift in cancer drug discovery. So this is our contact in the company. And last but not least, I wanted to, in this combination of the two worlds, thank, all the people involved in this incredible journey. Of course, the lab. I mean, the lab is still very much, active in this field. We have initiated this project with Alain Chariot back in the years, with whom we actually built the first conditional knockout, other collaborators at Giga. I wanted to highlight also here Francesca Rapino, who we worked together in the lab, and now we actually co-founded the company, and she took the lead in the company here in early 25. We have a lot of collaborators, of course, around us, and a fantastic team at THERATRAME that is really pushing this drug discovery incredibly well. So I would like to thank you for your attention, and I would be happy to answer any questions if any.
Thank you so much. The story is very inspiring, and it shows us that it really takes a village to take from an innovation from the lab to people's lives. So we are really very near to our timeline, to ending our timeline. But if you don't mind, I will ask you one question to each speaker, okay? So first, I want to thank you all for this very interesting session. And I want to follow up with Elodie. So my question is about for institutions that are looking to strengthen their IP management, what would be the top 2 practical steps that they should prioritize to build an effective patent portfolio?
Thank you. I would say it's important to have a good invention disclosure form with already the questions about the contractual aspect and so on, and to disseminate to the researchers the importance of filling the form as soon as possible, because by detecting as much inventions as possible, then you can then build on the patent portfolio. And yeah, also a big challenge is co-ownership, because we have many collaborations with institutions. So now what we have now is a standard co-ownership agreement that has been the result of a lot of experience, and so that should fasten the discussions with the partners before the patent filing.
Yes, and I do believe that just establishing very sturdy exploitation plans from the beginning to define the ownership of the key exploitable results of projects would also add up to that strategy. Thank you so much. And now for David. So if we considered the Europe's innovation dilemma, what specific policy changes or institutional incentives do you believe would most effectively encourage young researchers to engage more actively in knowledge transfer and also by patenting, of course?
Yeah, so I think, there are a couple of initiatives in that respect. I think, from the TTOs also, and it's across Europe, I would say, there's always this contradiction in which researchers, they have freedom to research, to choose their topic of research as far as they are recognized by the community, and it's according to quality standards. But on the other hand, there's more and more pressure exactly to create more startups, to create more revenue for, etc. And those two things are contradictory, let's say, to a certain extent. A researcher that has a very interesting technology and starts a collaboration every 6 months, it's very difficult to engage in a tech transfer pathway. So, and then it's always one of the things, you have the stick or you have the carrot. So which one you use? So we are in university, so we don't like to use the stick. One thing, it's also the question of create incentives for researchers. So the ones that wants to stay there. One very concrete thing is some institutions are doing it. For instance, University of Eindhoven in the Netherlands or the Curie Institute in France, they are creating positions that are in the France is called, I think it's like inventor, researcher or something like that. Positions that are just focused not on publishing, but on creating patents, creating spin-off, creating collaborations, and so on. I think that should be a way to go.
So thank you so much. And now the last question is for Pierre, it's on the Q&A box. So in your experience, what was the most challenging part of transitioning from fundamental research to product development?
It's a very tricky question because basically everything is challenging in this transition. So, but at the same time, it's very exciting. So I would say that one of the best advice is to be very much open-minded, to be listening to people, but at the same time to stay extremely strategic because you have many different options there, and what will make the difference in a project that is reaching the milestones is really that you're able to strategically pinpoint what is essential for your project and what would be the killing experiments or the killing recruitment or the killing aspects that would make this happen. And the last thing that is, everything is challenging. So you have to find people that would support you for real. So we got the chance from the early beginning to meet, extraordinary people that are not only scientists, but also entrepreneurs. And they were really supporting us in a completely, free manner, just because they were really wanting to support the project. And this has, an incredible value because then, you really have a couple of people around you to which you can, ask the stupid questions because, everything is new, everything is different. It's super exciting, but be clear on your strategy and be clear on your milestones because this is one of the main caveats for people transiting from the academia to the biotech is to lose yourself a little bit in all the things that needs to be done. So list the key points and then make sure that you recruit, you find the money and the people to achieve these milestones. I think that's the best advice.
Thank you so much. So we need to end our session. I want to thank everyone again. So to our expert speakers, thank you so much for taking your time to let us know what is happening in the world of IP management, of knowledge transfer. Thank you for the inspiring story also to Pierre. Thank you so much. So I just saw here. Okay, so we have some answered questions also in the Q&A box that you can check later also.
Okay.
So I believe that we must finish our session. So thank you all. Thank you to all the attendees that stayed until the end. It was a pleasure to host this session. Thank you.
Thanks for the invitation. Bye-bye.
Bye. Bye-bye.
Bye.